Hi guys, so we are moving on to something new today. We are going to talk about transcription. So don't forget, I have your note sheet. You might want to split your screen and put your Edpuzzle over here so that you can see it, and put your note sheet over there. There will be some questions at the end of the Edpuzzle, and they are questions that are on your note sheet. So you can do the note sheet first, do a little practice there. We'll go over how to do that in the lesson. And then just answer the questions on your Edpuzzle, and that's where I'll get your grade from. All right, so let's start with our presentation here. All right, so if you remember, and you just had a quiz over DNA and RNA, all right, we always start with the DNA, which is double-stranded, double helix, right? Stays in the nucleus, contains all the information for all of, it's like your blueprint, all the information for what makes you you. All right, in order to use our DNA, we are going to make RNA. And today we're going to be talking about the process of transcription. That's how we get from DNA to RNA, all right, as we do the process called transcription. Today we are actually going to be making mRNA. Remember, there are 3 types of RNA. So mRNA is messenger RNA, and that's the one we're going to be dealing with today. We won't have to deal with the other 2 types of DNA until we do translation, which is the second process where you take the RNA and make it into a protein. All right, so make sure you fill in those 3 blanks at the top of your note sheet. Remember, we start with our DNA, right? Through transcription, we get to RNA, and through translation, we make proteins. All right, so for today, we are just going to work on this first piece, right, from DNA to RNA transcription. All right, so why do we need to do transcription? Okay, so our problem. DNA contains all of our instructions, all of our directions to make proteins, all right? But proteins are made attached to ribosomes. If you remember, that's an organelle in the cell, all right? And the ribosomes make proteins. They are floating around in the cytoplasm, all right, or stuck to the rough endoplasmic reticulum, the rough ER in the cell, all right? But here's our problem. Where is the DNA? Right? The DNA cannot leave the nucleus. The DNA is too big. It's stuck inside the nucleus. So to get those directions, instructions out of the nucleus, we're going to change the DNA into RNA. And that's why we need the process of transcription. All right, so transcription is our process that takes the information in the DNA and copies it into messenger RNA, because the messenger RNA can then leave the nucleus to do the process of translation out in the cytoplasm. That is what we will talk about next class. So today we are just focusing on this top piece. All right, so the reason we need transcription is to make the message in the DNA able to leave the nucleus. So we make RNA that can leave the nucleus. It then travels out to the cytoplasm, to the ribosomes to deliver the directions and make a protein. All right, so we have a couple of steps here, and I'm just going to go through the steps with words first, and then I've got pictures on my next slide for you. All right, so transcription, going from DNA to RNA. Step 1, we have to unzip the DNA, right? DNA is double-stranded, like you see here. So we need to unzip it up the middle, make it single-stranded. We do that with an enzyme called helicase, right? It unzips the helix. Once the DNA is unzipped, we're going to use another enzyme. This is called RNA polymerase. Maybe when we did the Edpuzzle on DNA replication, it probably talked about DNA polymerase. That helped make DNA. So RNA polymerase is an enzyme that's going to help you make RNA. So it's going to add complementary bases, right? Remember, RNA has A, adenine, G, guanine, C, cytosine, or U, uracil. This is RNA. We don't have any thymine. All right, so we're going to add those bases that are complementary. All right, we'll go over what that means again in a minute when we do our example. To make the RNA, and we always go in the same direction. It's from the 5 prime, that's called a prime in this case, 5 prime to 3 prime end. So we always go from 5 to 3, and I'll show you that when we do our example. All right, step 3. The mRNA, the messenger RNA, is made. It's going to leave the nucleus, travel out to the cytoplasm, and find a ribosome to attach to. So we're going to travel to a ribosome out in the cytoplasm. All right, from there, kind of getting ready to start the next process, and something important for you to know that we'll go over more. When you read mRNA, you're going to read what's called a codon. So you're going to read it 3 bases, 3 letters at a time. Right? Each 3 letters are going to code for a specific amino acid, and that's going to help us in the next process that we talk about next class. All right, but you got to get used to reading the mRNA 3 bases at a time. That's a codon. All right, so here's my pictures. So step 1, right? I have to unzip the DNA double helix. So here's my DNA on the outside. It has T's in it. All right, these bases are complementary, right? T goes with A in DNA. So we unzip it. So the helicase is going to unzip it. Step 2, we're going to use RNA polymerase to start making complementary bases of RNA. So here's my RNA strand, right? A goes with U, C goes with G. That's complementary. So my RNA polymerase is going to move along this sugar phosphate backbone, move along the side of the RNA, right, and add those complementary bases. All right, once it reaches the end, it'll be released, and the RNA is small enough to leave the nucleus and go out into the cytoplasm. It'll find a ribosome, and from them we'll get ready to read it to make the instructions into a protein through the process of translation, which we'll talk more about next class. Right, but here's my mRNA that left the nucleus, right? You see my bases, A, U, A. There's a G, there's a C. All right, this blobby looking thing that looks kind of like a rock, right? That is my ribosome. So our mRNA is going to slide through the ribosome, and we're going to read it 1 codon at a time. So basically 3 bases at a time, a codon. All right, so here we go. We'll do a little practice here. So we're going to complete the process of transcription in the appropriate direction. So here is my DNA strand. We can pretend it's already unzipped by the helicase. So basically, we're going to be the RNA polymerase. We're going to come in and add the complementary bases, but we have to go in the right direction. All right, so the right direction is always from 5 to 3 prime. So in this case, I am going to go from this side, 5 prime, towards the 3 prime end, if I were doing this in reality. All right, so I'm making RNA. Keep that in mind with what bases you need to use. All right, so if I have A, the base that is complementary to in mRNA to A is U. All right, so I did put this picture on your note sheet if you want to fill it in. I don't think you can type on the picture, so I would just put a little blank underneath it to type your letters. All right, so A goes with U. All right, C is going to go with G. All right, what was the little saying? It doesn't work for the AU, but C, car, garage. All right, A is going to go with U, uracil. G, garage, goes with car. All right, again, C goes with G. C goes with always G. Okay, in RNA, we're having A, so that's going to go with A, U. T is going to go with A. If you remember the little saying from before, apples in the tree, so it works there, but just make sure that your RNA never has T's in it, because that's not possible, right? It always has U's. And last but not least, G goes with C. All right, so that is my transcription. I made my complementary RNA that can now leave the nucleus and go out into the cytoplasm. All right, but last thing, let's talk about codons. So how many codons are there in this RNA strand? All right, so codons, remember how many bases at a time? 3. All right, so if I read it 3 bases at a time, how many codons do I have? 1, because I have 1, 2, 3 bases. 1, 2, 3 bases. 1, 2, 3 bases. All right, so in this strand of mRNA, I have 1, 2, 3 codons because I have to read in groups of 3. All right, that is the end of my PowerPoint for you. All right, what you probably want to do now is on your note sheet, right? Do the practice first. Try it out. Just pause your ed puzzle. You can always go back to it. If you're really competent, you could try doing it without, right? But answer these questions, right? Do what we just did. A goes with what? C goes with what? Make the mRNA. I ask you to highlight the codons, so you could use the little highlight tool and just like highlight and then maybe pick a different color, highlight the next one, different color, highlight the next one, right? And count how many codons there are, right? And then I gave you another DNA strand down here. Make it into RNA, highlight the codons, how many codons, all right? So those are going to be questions at the end of the Ed Puzzle video for you to answer to get your grade for this assignment, along with any other questions I ask you. All right, guys, hope you have a great day. Only a few more days left.